Good afternoon, and welcome to this special installment of the MIT Club of Boston seminar series, The Science of Making and Saving Memories. I'm Hyuna Park, one of the directors of the club. It's now my privilege to introduce our speakers. Li-Huei Tsai is the Pickauer Professor of Neuroscience in the Department of Brain and Cognitive Science at MIT and the director of the Pickauer Institute for Learning and Memory and the Aging Brain Initiative. Her pioneering work on unraveling the mechanisms involved in Alzheimer's has led to several novel therapeutic strategies. Dr. Tsai is the recipient of too many awards to mention, including the prestigious Lifetime Achievement Award from the Society of Neuroscience. Steve Ramirez is a professor at Boston University's Center for Memory and Brain Center for Systems Neuroscience. He received his PhD in neuroscience at MIT in 2017 in Susumu Tonegawa's lab, where he and a colleague broke ground by figuring out how to manipulate memories in mice optogenetically. Dr. Ramirez's hope is that insights from this basic research will point the way to new treatments for a range of diseases involving memory, such as PTSD, depression, and addiction. Lauren Aguirre graduated from MIT in 1986 with a degree in science writing and Russian. She began working at the PBS series Nova for Europe and went on to become a producer, science editor, and the director of digital media. She left Nova in 2017 to write The Memory Thief and the Secrets Behind How We Remember, a Medical Mystery, which won the 2021 American Book Fest Science Award. We've set aside 15 minutes for questions at the end of the event for Li-Huei, Steve, and Lauren, so please go ahead and put your questions in the Q&A box at any time during this session. If the question is for a specific person, let us know in your entry. Thank you. So now I'll turn it over to Lauren.
Hi, everybody. Thanks for coming. I'm going to divide this into 4 sections. So first, we'll start by speaking with Steve about the basic science of memory. Then we'll speak with Li-Huei about her research in Alzheimer's disease. Then I'll facilitate a conversation between the two of them about how science advances, how medical science advances are, what the opportunities are, and then we'll take the questions at the end. So if we could start by putting up the beautiful picture that Steve sent me that was made in his lab. And Steve, can you just tell us what we're looking at here?
Yeah. First, thanks for having us here today. This is awesome, and it's very heartening to see how many people care about memory. This is great. So what we're looking at here is it's a small cross section. So it's about 50 microns thick. So it's thinner than a nail of an area of the brain called the hippocampus that we know is important for learning and memory. And specifically, what you're looking at is the green brain cells are ones that we think are involved in the formation of particular memories in the brain. So I love this image because memory, for the most part, doesn't feel like something that we should be able to feel and touch, right? It kind of has those ephemeral qualities to them. And yet, if you have a good enough microscope and good enough genetic tools, we can zoom in and actually start to image the physical basis of memory itself. So I love this because whatever memory is, this is at least a very small cross section of it as one's being formed in the brain.
So what is the memory of in this case?
Yeah, so in this case, we work with 2 different kinds of memories in rodents in particular, so positive and negative memories. In this case, it's the memory of an animal being fear conditioned. So this is just a standard task that we use in the lab to give animals an aversive experience so that we can see how those memories are formed. And ideally, with this picture in particular, is to go in and try to artificially manipulate those kinds of memories. And you could imagine using animal models of PTSD and anxiety, for example, and trying to prevent the return of these kinds of aversive experiences. So that's at least what we're looking at here in particular.
Cool. All right, if you can take that picture down. So can you backtrack a little bit, Steve, to the work you did when you were at MIT, which sort of launched a new field in a way, and just tell us briefly, if you can, what those two experiments were.
Oh yeah, it was awesome. Those were like roller coaster years in the best way because I could give you every hypothesis like known to the field about why it's important to try to isolate the physical basis of memory. But when we were doing these experiments, it was just fun. It sounded like sci-fi. The therapeutic potential was kind of through the roof. So we thought we inherently gravitated towards these kinds of experiments. So the first experiment, the idea was, could we find sets of cells in particular that were involved in forming just one particular memory? And if we could find those cells, was there any way of tricking them to glow, for instance, so that we could visualize them under a microscope? The brain is normally kind of pink and custardy, so we need things to glow green or blue or red or some of these colors so that we can go in and take pictures of them. So we use these tools that help us find the cells that held on to one particular memory and then trick those cells to glow green like we saw in the previous image so that we could visualize it. But then the next step was, could we go in and artificially tinker with those cells and turn them on and off to see if that would be enough to actually stimulate that memory, to re-stimulate it. So cues do this in the world to us all the time, right? Like you could be walking down Boylston Street, you'll smell a bakery and it might remind you of like I don't know, like a bake fair or something from high school. And it's a small little cue that triggered the world of a memory that comes back online in our brain. So we wanted to do this from within the brain instead, like kind of like a domino set. Like could we knock over one domino in the brain by activating these brain cells that held onto a memory? And would that kind of cause this cascade or this chain reaction of recollection in a sense? So that was our first big discovery, was that by stimulating or turning on these brain cells that we think were holding on to a memory, this seemed to be enough to actually reactivate the expression of that memory in particular. So that was kind of the first series of findings that that helped us go in and start to artificially turn these things on and off in the brain.
So you're able to change memories too, which we actually know, or science has found, that happens naturally all the time, which is a little upsetting to think that your most cherished memories might not be quite what they were. And in fact, maybe the more you remember them, the less like the original they were. But is there also kind of an opportunity there therapeutically?
Yeah, absolutely. I mean, one thing that we've learned from the past like half a century of research is that it's kind of counterintuitive, but memory, as far as we know, isn't like an iPhone video of the past where we're taking a video and then we're just replaying it and replaying it each time we recall it. It seems to be much more dynamic and much more malleable in a sense. So each time we recall a memory, for instance, it's thought that it's constantly being warped and updated with bits and pieces of information. That flexibility can be a good thing. I mean, there's some leading theories that this might be why we can imagine ourselves in the future and actually are the creative species that we are, for instance. So memory's malleability can be a good thing because on the one hand, it gives us almost like a flexible scaffold for recalling our past. But then from the therapeutic perspective, some of the work that we've been focusing on for the last decade really is taking advantage of this idea that we can update memory with bits and pieces of information, because you could imagine artificially tinkering with an aversive experience to make it less aversive, for example, to try to mitigate symptoms associated with anxiety or PTSD, or the converse of that, where we could go in and try to boost up the emotional oomph, in a sense, of a positive experience to see if that kind of imbuing a memory with positivity can help ameliorate symptoms associated with depression, for example, and anxiety as well. So we think that that flexible nature of memory can be used in a therapeutic manner, either invasively in rodents by going in and optogenetically, again, playing around with these brain areas, or non-invasively in humans with different kinds of therapies and cognitive behavioral approaches that leverage this flexible nature of memory, which we think can be a bit of a silver lining in an otherwise strange phenomenon.
Is there something adaptive about the malleability of memories and also the fact that we forget memories? Maybe start with the malleability of memories first.
Yeah, so it's, I mean, this is a hot button today in the field, like this idea of why would evolution favor a system that can be written and rewritten in terms of the contents of its past, for example. Now, one example, at least that I like, is like we study the hippocampus. We know it's important for forming memories, but we also know it's important for projecting ourselves into the future, for example, like imagining future scenarios. So at least one idea here is that by having the same circuitry for the most part that's involved in recalling the past be involved in imagining ourselves in the future, when we imagine ourselves in the future, we are using our previous experience to help guide that decision. For example, did we like the restaurant that we went to or not? Did something good or bad happen? Even if we haven't encountered that exact experience before. So for example, I taught in person this semester, and I've been into plenty of classrooms in my life, but I've never been into a classroom, for instance, while the world was only partly spinning and the pandemic was going on. And yet I could imagine myself in that scenario from multiple different angles with a bunch of different sort of what if scenarios of what might happen in the classroom. And it's not that being able to project myself into that future scenario that I haven't experienced yet does at least partly tap into these repositories of my previous experiences of having gone into classrooms before. I know the dynamic of what it's like having dozens of students in a room and things like that. So we think that at least that memory at least enables, one of the things it might be able to do is enable ourselves to project ourselves into the future, for instance, and that requires this kind of malleability or flexibility or creativity, I guess, to put it another way, to be able to imagine ourselves in scenarios that we haven't yet been a part of before.
So we looked at a little teeny tiny slice of the hippocampus at the beginning. You know, sort of very generally speaking, people talk about that as the memory center of the brain. In my book, that memory center, the hippocampus, gets severely damaged by people who overdose on fentanyl. So can you tell us what we know now about the role of the hippocampus in memory?
Yeah, I think one thing that we've learned is we know it's important for different kinds of memories, especially forming and recalling memories of our experienced past, for example. The way I think about it, though, is that when we think of a memory, there's sights and sounds and smells and emotions and contextual temporal information embedded into that experience. And we think that recruits parts of the brain that are involved in processing the sights and sounds and smells and temporal, contextual, emotional information of a memory too. And that recruits underlying circuitry that's distributed throughout the brain in 3 dimensions, like high and low, left and right. So at least I think of memory as this otherwise distributed phenomenon that exists in 3 dimensions in the brain, and the hippocampus is one of those areas, maybe like a conductor in a sense, that's involved in stringing together these sights and sounds and smells into a cohesive whole that we experience. And in that sense, we tend to experience kind of cohesively, unless like a Picasso, for example. So at least the 2 things are that the hippocampus might be involved in indexing these different processes of memory, but the second thing being that memory otherwise is a phenomenon that exists distributed throughout the entirety of the brain, in areas like the hippocampus are sort of like key centers there that are involved in stringing that activity together as well. Maybe like a conductor of an orchestra as an example.
So a memory is really a pattern of activity of connected neurons activated together at in a certain sequence. Let's say you had a memory of an event and someone could go in with a laser and sort of remove all of those connections. Would the memory truly be gone, or would there be a way to create that same pattern again of activity?
So this is a fantastic question, and maybe this is just like my hot take of the day, but we don't know the answer to that, I don't think, because there's a couple of scenarios here, which is if you think of memory as a book in a library, and let's say something happens, you erase those brain cells, or maybe a neurodegenerative disorder happens, for instance, and we can't recall a particular experience anymore. Is it because the book of the library burned down, right, and that information is no longer there? Or is the librarian kind of on a break and the books are still there, but what we need to access those books has become impaired, for instance? It's a pretty fascinating, I think, line of research recently, because more and more evidence, at least, is partly suggesting that those books are pretty hard to burn down. And it's a lot easier to maybe break down mechanisms where the librarian seemingly just is on a break, more so than actually breaking down the books themselves. Because as long as the brain is there, for instance, memory does seem to be pretty amazingly resilient in terms of how we can potentially even bring it back. So there's a slew of studies that have come out, especially in the past decade and a half, that have suggested that memories thought to be lost to different kinds of amnesia might still be retrievable, but not through natural circumstances. Like we need these kinds of contemporary tools of neuroscience to go into the brain and start probing around and forcing that memory to be retrieved. So we're sort of forcing the brain to, or we're using something from the outside to fix that aspect of the brain, for instance. So it's unanswered there in terms of the forgetting component, or whether how much forgetting even happens, or especially in light of amnesia. But I'm a very cup is like 3/4 full person with this, and I think that memory, it might be a little bit harder to break than we previously imagined. But given the tools that we have now, at least we might be able to go in and try to artificially retrieve it.
Well, that's good news. That's a good cue to go over to Li-Huei to talk about retrieving memories. So I first heard about your work actually at a MIT Better World event in 2017. And you gave a talk about gamma entrainment, and I thought, oh my gosh, this is the right time to be writing a book. This is very exciting. So can you tell us what gamma entrainment is?
So I think that most of you have heard of brain waves or brain oscillations. So, I have to be honest, for the longest time, I think even neuroscientists didn't know what it meant. But, I would say that we've slowly come to appreciate its purpose. So, so the brain generates waves or oscillations to transmit thoughts and information by coordinating the activity of nerve cells or neurons. So when I say entrainment, I really mean using different stimulation conditions to make the nerve cells in the brain to generate these waves and oscillations.
So how would, so the gamma entrainment though is sort of externally strengthening gamma waves inside the brain with light or sound oscillating at that 40 times a second. And your lab has found that appears to improve memory in mice and get rid of some of the signs of Alzheimer's disease. And in mice, you do it optogenetically, obviously, or that was the first experiment. Obviously, you can't do that in people.
Right.
But it sounds like pretty crazy. And what research led you to that idea that could work?
Ooh. So first of all, there is a body of literature reporting that gamma waves, or the power of gamma oscillations, is compromised in Alzheimer's dementia. There's some reports in human subjects, and there are a lot of reports in various animal models of Alzheimer's disease, including the amyloid mouse models, the tau tango mouse model, and even in the humanized APOE4, which is a major Alzheimer's risk gene mouse model. So it seems that this is something common in Alzheimer's disease. And we actually also tested in our own hands, and we found that even in the pre-symptomatic stage of the disease, in the mouse model, before we observed profound amyloid plaques develop in the brain, in the hippocampus, as you guys just discussed, the power of gamma oscillations is already significantly reduced in the disease model compared to the litter-mate healthy animals. So obviously, the next question we had was whether, if we boost the power of gamma in the brain, whether, it will have any measurable effects since this happens so early. So we thought that it may play a driving role in either neurodegeneration or cognitive impairment. So, so the first experiment, as you mentioned, we used the so-called optogenetics, which is very invasive. You need to use a viral vector to introduce some sort of opsin into a particular region of the brain, and then you need to implant fiber optics to the brain, and then use a laser to drive the neurons. But in 2009, my lab actually collaborated with Chris Moore, at the time an investigator at MIT, and we demonstrated that using optogenetics to activate a very specific population of neurons known as the POP albumin positive fast spiking interneurons in mice can very effectively boost gamma oscillation power in the brain. So, with that knowledge and, we know how to do it. So the first experiment, we decided to use optogenetics to boost gamma in the amyloid mouse model.
Is there any understanding of the mechanism, like how that works, how that might clear amyloid beta or tau tangles, which for the audience that doesn't know this, those are the two signature toxic proteins that accumulate in the brain during Alzheimer's disease?
True. So, so the signature pathology of Alzheimer's is the very abundant amyloid, or known as senile plaques, as well as neurofibrillary tau tangles in the brain. So we show in our first publication using either optogenetics or using light to stimulate the brain, we show that we could boost gamma power, and then that was associated with a very significant reduction of beta amyloid. So obviously, there were a lot of questions about how does this happen, and I can tell you, in the last, I would say, several years, this really has been the question that my lab has been focusing on. So the question is, so the bottom line is, very, very surprisingly, increased gamma power in the brain. So basically meaning, you generate this rhythm, this nerve cells, they fire in a very rhythmic way, not only affect the physiology of neurons or nerve cells, but actually also elicit response basically from all the other cell types in the brain. This includes, microglia, which are known to be the innate immune cells and very important for clearance of toxic waste in the brain, and astrocytes and the vasculature, very importantly, and activation of vasculature is also known to mediate clearance of toxic waste. So the bottom line is, very surprisingly, increased gamma power in the brain elicits a holistic systems level response that benefits the brain.
You have so many other interesting projects going on in your lab. I wanted to talk about one paper that came out recently. So, we've all heard about, if you want to sort of reduce your risk for developing Alzheimer's or other dementias, use your brain, exercise your brain. And I don't think it was clear what the mechanism involved was. There's two concepts. One is cognitive reserve, sort of if you have a good education, you have a sort of a bank account that you can draw from, even if there's pathology building up in the background. And then cognitive resilience, that somehow your brain can be resilient to damage. So, but your lab recently found a genetic connection with that. Can you talk about that a little bit?
Yeah. Thank you for bringing this up. So it has always been a fascinating phenomenon, because most people, if you see as they age, their brain accumulates more and more, more and more abundant pathology, plaques and tangles and all kinds of other damage, and then that absolutely associates with cognitive decline and eventually dementia. However, if you look closer, you will always find exceptions. There are individuals, despite their brains full of those pathology, and yet they are, they're still very normal. They're lucid. They don't, lose their memory. So, so we specifically focus on those individuals and really wanted to know, why are they different? So, so we work with two large, brain banks. Those were, longitudinal studies. People recruit individuals, from middle of their life and follow them all the way until they die, and then have the medical history and also their biological brain tissues. So we compare their the signature of their of their brain with those individuals who who are normal, who are susceptible to cognitive decline and dementia. And then we found that those resilient brains, they have increased gene expression signature associated with synaptic function, GABAergic neurotransmission, and stronger mitochondria function, and so on. But most importantly, with this kind of artificial intelligence, machine learning kind of big data analysis, we are able to further identify the master regulators of the expression of those genes. And we were able to eventually identify a family of transcription factors known as MAF2 transcription factors that their own expression levels, as well as the genes regulated by them, they particularly show good correlation with cognitive resilience.
So hopefully our MEF2 genes are all being expressed right now. So I think I want to move on now to a discussion between Steve and Li-Huei, how we can turn these great insights in the lab into treatments for memory disorders, particularly Alzheimer's disease, which, as many of us know, has been I think you described it, Li-Huei, as a humbling experience being an Alzheimer's researcher, because despite many decades and lots of hard work, there is yet to be a truly effective treatment. So I wonder why is it, Li-Huei, that it's been so hard to come up with a treatment for Alzheimer's disease?
I think we all have to agree this disease is extremely complex. And from the first appearance of pathology to the appearance of symptoms, there can be decades in between. So at the cellular, molecular, and biochemical levels, this could mean that there are numerous events that can happen in between. So, in the end, it's a question of what will be the most effective target to bring back the memory, okay? So that's the first thing I want to say. And second thing also, I also mentioned in my recent Boston Globe op-ed article, we really don't know, whether Alzheimer's disease is a single disease or, it's, a collection of different diseases, meaning people get Alzheimer's, from different etiology. And if this is the case, we probably need to first know how to stratify Alzheimer's patients. And then we can develop effective treatment against a different specific subpopulation of patients. And I would say that I think more and more people will accept this notion that, people probably get Alzheimer's disease from different etiology. But we are really at the very beginning of trying to figure out then how do we, target these different populations of patients.
Mm-hmm. So going back to sort of the idea of the importance of basic science that might be kind of agnostic as to, it's not translational. It's not seeking to solve a problem or come up with a therapy. For example, CRISPR, was initially having to do with understanding the bacterial immune system, and now it's an incredible tool. So there's an argument for just, fundamental curiosity-driven science. But you, Steve, mentioned early on that you specifically are interested in fundamental science from a therapeutic perspective. And I wonder if you can tell us where that came from, and if you think there's any sort of downside to having a goal in mind when you're developing a scientific question.
Yeah, I mean, I still consider myself like a basic scientist at heart with these things. And the way I think about this is, like the history of science is riddled with so many different examples of studying one particular thing that a discovery happens and then it gets refurbished for something else like 100 years later. So I think that the basic premise here is, in science, I think knowing is better than not knowing, and whether that knowledge is going to be applicable tomorrow or whether it's going to be applicable in 100 years depends maybe on just what kind of career you want, right? So one example that's used often is, let's say you're Einstein, right? And you care about the physics of the world, and you're tasked with figuring out stuff about how objects warp space-time. You figure it out, you have your theories of relativity, fantastic. Now, 100 years later, if anyone's used Google Maps on their phone, you need a signal to go from here to an antenna to a satellite back to Earth, and it has to take the curvature of space-time into account, or GPS. We wouldn't have GPS, basically. Now, Einstein didn't necessarily care about ending up in everyone's back pocket like as an app in your iPhone, right? I wanted to figure out stuff about the physics of the world. And then that was applied in a way that gave us GPS and other game-changing technologies. So I think that it could be the same thing with neuroscience, where 80s, 90s, you have scientists that are studying algae in California ponds, and they find a protein that's sensitive to light. And then that protein a couple of decades later is engineered into neural tissue, and bam, you get optogenetics. So I think that there's good reason for wanting to invest in basic science, because again, the premise here is knowing is better than not knowing, and the applications there are going to happen. It's a matter of when, not a matter of if. So when we study things in lab, projects tend to fall under these two camps. There's we want to know how memory works, and we want to know how the brain works, and what this just tells us about the organ that is the brain. And that's about half the projects. And then the other half are, we can have a goal in mind, like there's a particular symptom associated with a particular neurodegenerative disorder, and let's try to reverse that particular symptom and see if it might be more inherently translatable, for sure. So we can be greedy about these things and go have both lines of research, where some are basic science at heart and some are inherently more translatable at heart. I do think we need both simultaneously, but I think that we need a bigger rallying call for the basic science aspect of it all, because I think that until we have like some fundamentals about how the brain works, or some ground truth principles about the brain, which we're going to need basic science for that. It's like, until we have that, then I think that the problems remain like otherwise pretty challenging. So I think we need basic science for that reason.
I cannot agree more with Steve. If I could just chime in a little bit, brain oscillations cannot be more fundamental in the beginning. When physiologists, they were able to detect that there are electrical signal that they can detect in the brain, right? It was so fundamental, right? And for the longest time, as I said, neuroscientists were fascinated by this kind of signal. No one really know what does it mean or anything. And then we later on, we actually use optogenetics, again, extremely fundamental, basic approach to try to manipulate these brain waves. And without any of this knowledge, it wouldn't be possible for us to now one day think about maybe, hey, we could translate this into something beneficial for a particular brain disease. So I cannot agree more that fundamental basic research is really the key to everything.
What about big science? I'm thinking of the Brain Initiative, which is maybe $6 billion by the time it's said and done. Is that money well spent? I've heard arguments on both sides, and that's for either of you, that question.
Yeah, the short of it is I do unequivocally think it's money well spent. Whether it can be spent better or more effectively or efficiently, sure, I think we can always do that. And as we accrue more knowledge about how to, Li-Huei had a fantastic point on how we even define something as complex as Alzheimer's disease, and Alzheimer's might be an umbrella term for multiple pathologies, for instance. And we can say the same thing about depression and anxiety, that these might be umbrella terms that we're using for a medley of pathologies that are brewing underneath. So the more we refine that criteria of what these really are, then we can think about what the next big wave of funding might look like what we're going to invest in. It's no coincidence that investing right now in big science, in machine learning, for instance, or with the NIH's various programs for redefining psychiatric disorders from the brain's perspective, have really picked off, because I think that they've made a lot of headway, and that's great. We can definitely iterate and refine how we end up investing in these big picture ideas as we learn more along the way. So money well spent, yes, and it'll hopefully continue to be money more effectively spent as we just accrue more knowledge about how the brain works.
I cannot agree more. With this Brain Initiative, since the Brain Initiative project took off, I think it really accelerated our fundamental understanding of the brain at multiple levels. We realize that, there are new type of cells, and, why the different brain regions have different function and so on. And more recently, I think the Brain Initiative really supported the kind of big project like, computational genomics and artificial intelligence. And in the disease point of view, it started to unravel the polygenic risk of human diseases, not just Alzheimer's, almost all the brain diseases, depression, schizophrenia, and so on. They're all polygenic. People may have a combination of different risk gene profile, and which would, in a way influence their risk of developing a particular disease. So I think without the big investment through Brain Initiative, the progress would have been, we wouldn't have made this kind of progress.
So Li-Huei, you explained why it's so hard to understand Alzheimer's and find treatments just because of the nature of the disease, and it's a disease of aging. But are there any other reasons you can think of why it's been so hard to translate what scientists do know into viable treatments in people? If you could wave a magic wand and change something else about how the system works or how things are funded, what would that be?
Yeah, so, for something, this complex, I would like to borrow a phrase from my MIT colleague, Professor Andrew Lo. He has this very famous essay known as Valley of Death. And, this really can apply to drug discovery for any human diseases, especially for complex of disease. Multiple shots on goal really is the way to go. For Alzheimer's, I think, everybody knows that, for many years, we have had very, very few targets. And if that, one or two targets don't work out, I mean, we put ourselves in a very difficult situation. So, I mean, I know, I don't want to sound greedy. I know that, there are already investment, like brain initiative, as, you just mentioned, but, we really need to invest more so we can have multiple shots on goal, to really, in the shortest time frame to come up with something that actually gonna work. So that's the first thing. And second thing, I think people may want to think more about using different modalities, multimodal kind of approach to treat complex diseases. In cancer, people use a cocktail of different drugs, and for certain infectious disease as well, such as HIV infection. And I think for complex diseases like Alzheimer's, this is something we really need to think about, seriously about.
So I have a million more questions for both of you, but I think our audience probably does too. And we promised we would take questions. So I think this would be a good time to turn it over. And Hyana has been looking through the Google Doc of questions that are coming in. And I have not been looking at them because I've been paying attention to this. So Hyana, if you want to just go ahead and put out questions that are in there. And if someone has said who it's for, you can point it to them. And if not, we'll just try to figure out who's the best position to answer the question.
Thanks, Lauren. What a fantastic session. And we have a lot of questions. But before I turn to the Q&A from the audience, I wanted to ask Lauren, who has been researching this topic for her book as a science journalist, not as a neuroscientist, what your kind of observation of where are we today in our understanding of the brain and our ability to actually find solutions for the ailments that people are experiencing both themselves or in their families.
Yikes. I guess I would say one thing that really struck me as I began is sort of how depressing this field is in a way, not the basic memory field, but the Alzheimer's field because of the failures. But then, the more people I talked to, the more I kept hearing, the words optimism. Yes, it's humbling being a scientist. I asked someone, don't you get depressed doing this failure after failure? And he's like, no, being a scientist is fundamentally an optimistic choice because you have to believe that you're going to be able to answer the question eventually. And if not you, then the people who come after you.
Yes.
So that was what I came away with. Is it's always more complicated than you think. There is no hammer of truth. I think you used that phrase with me, Steve. Like it's always, how do we know what we think we know? Because people get tripped up. But it's just a great adventure, and I am hopeful for the future.
Thank you for that. All right, the first question is for Steve. Do you know what or where in the brain contributes to good or poor memory in people?
Yeah, so I guess there's two different things here. There's having good memory, and then there's good kinds of memory, so like positive memories and experiences. So I assume this means the former. For having a good memory or having a poor memory, there's a lot of psychological tricks for trying to encode information efficiently, and it's kind of tough because for anyone interested, there's a great book, Moonwalking with Einstein, that kind of covers this. And there's this idea of building a memory palace, they call it, for example, where you can learn different kinds of mnemonics to learn to remember like 2 decks of cards in under 2 minutes or something like that. And it's a great trick, but then how translatable is that, right? It's like, it may not give us photographic memory per se. It's just a trick that takes advantage that our spatial memories tend to be pretty remarkably good in some instances. But I'd say that if there's no silver bullet yet, that the things for having a good memory tend to be the things that we don't get enough of and the things that no one likes doing. So this would be exercise, good night's sleep, right? Don't smoke, like have a socially enriched lifestyle, which is harder and harder these days, right? So it's like the things like exercise, good night's sleep, social enrichment, having that kind of multi-pronged approach, at least collectively, would be good, both for memory, but also for cardiovascular disorder and for basically the rest of the body, right? It's like we're this unified organism where the brain doesn't exist just independent of the rest of the body. There's a lot of two-way channels of communication here. So for good memory, if we're trying to have good memory, I'd say that those tend to be at least some of the some of the more often quoted like temporary solutions for how to at least enhance or maintain a good memory.
Thank you. Next question is for everyone. How readily have you been able to track memory changes over time?
Is this a question for me or for Steve?
It wasn't targeted to anyone specifically. So would you like to start, Li-Huei?
Do I want to start? Sure. So if we say this is, if we track memory in people, there are quite a few things we can do. There are, a battery of cognitive tests one can use to evaluate memory function, all kinds of different memory, working memory, semantic memory, motor memory, all kinds of memories. And there are also different brain imaging methods, such as functional MRI, to measure connectivity between different regions of the brain, which a lot of times serve as a good proxy to look at how well your synaptic function is still, remain, and your brain can, efficiently process information to different regions. So that's in humans, and in animals, similarly. There are, battery of different behavioral tasks, as Steve mentioned, tasks to measure fear memories using, the fear conditioning paradigm, spatial memory using water maze or circular maze, bar maze, and also tests for working memory, short-term memories, and so on. So so memory can be measured in both experimental animals and in human subjects. Steve, do you want to?
Yeah. I mean, to echo that, from the tools perspective, like the things available these days are pretty extraordinary. I mean, from the idea of being able to go and image a brain intact while an animal performs a complex array of tasks, can measure anything from single cells all the way to 1 million, it's being reported recently. So we can really go and eavesdrop on this constellation of activity that's happening in the brain in real time using some of these contemporary techniques. And then in humans, of course, some of the tools like self-reports or fMRI and so on are some of our current best bets for being able to keep track of memories and how they evolve over time as well, or when recalled.
Mm-hmm. The next question is for Li-Huei. It's I'm wondering how early Alzheimer's disease starts. Is there any research on how early gamma oscillations are reduced in the different animal models?
Okay, so how early Alzheimer's disease starts? In humans, I think no one really knows, I want to say, because nowadays, Alzheimer's disease has a new definition, I want to say. So, people will consider to have Alzheimer's disease when they can detect beta amyloid plaque accumulation in the brain using, PET imaging, for instance. So Alzheimer's disease at this point is very much defined by the development of pathology. So if one develop pathology, but then also develop symptoms, then this is called Alzheimer's dementia. So I think there could be a very long time, as I said, between the detection of the amyloid pathology accumulation in the brain to the development of clinical cognitive decline. It could be easily 2 decades even. So if we want to say when can Alzheimer's start, it actually can start pretty early. And then there are also this another kind of Alzheimer's. So we're already talking about at least 2 different kinds of Alzheimer's. So what I just said is referring to the so-called late onset sporadic Alzheimer's disease. But then there are also the so-called familial early onset Alzheimer's disease. And these individuals carry certain, faulty gene products that they can develop Alzheimer's dementia even in their 30s. So in a way, even though Alzheimer's disease is an aging disease, but in certain individuals, it can develop pretty early.
All right. Next question is for Steve. What could be the evolutionary reason for forgetting?
Ooh, so this is a great question. And, one answer that's given that I don't know how much I buy for this is like this idea that we have areas like the hippocampus that are temporary repositories for information, and the hippocampus will send off memories as they age to different parts of the brain, for instance, and kind of stores them permanently there, clears up more room for the hippocampus to be involved in making new memories, for that matter. Now, we might be underestimating sort of the computational power of what 86 billion brain cells actually do here, because one extreme hypothesis would be that we don't forget, and that most of what we experience leaves some kind of measurable physical imprint in the brain. It's just that we don't need to revisit every bit of detail that we've experienced throughout our lives. We tend to remember what we had for dinner last night, or maybe what we did last Friday. But then if I was to ask, like, what did you do exactly 23 days ago at 2 p.m., you kind of have to think about it a little bit more and maybe use Google Calendar to figure it out. So it might just be efficient information processing, that we tend to access the memories that make it more likely that we can answer the task at hand, for instance. And then if we really dig deeper, or through artificial means like some of our work, and try to just bring back stuff that was thought to be gone, for that matter, it might at least hint at the process of forgetting. It's not to say that forgetting doesn't happen, but how robustly it happens, I think, is really this kind of outstanding question in the field right now. But let's just assume that forgetting happens in the brain, and one at least theory for why we forget could be, again, so that we don't have this kind of neural information overload, so to speak, so that we tend to select what's the most relevant aspects of a given experience, or what we're going to recall, and bring those back to our mind's eye, as opposed to every single detail of what we've experienced, which would, in a sense, like, taken to its logical extreme, if we were to recall every bit of detail of what we experienced in the past, we would be hallucinating now, as opposed to recalling a memory. And so these two things serve clearly very different functions, but at least that's one theory, is that we're selecting for what will help us solve this task at hand right now. That doesn't require every bit of our past experience. So fine-tuning that information over time means trimming some of the fat over time as well.
Okay, I think this is going to be our final question. Let's see. Okay, in a computer with core memory, the ability to remember the number 101 is accomplished by 3 ferromagnetic loops, 2 oriented one way and 1 oriented the other. What is the equivalent in brain memory? Who
I
wants to
guess
start?
yeah. So I guess this is a matter of answering how memory is stored in the brain as opposed to how memory is stored in a computer, for example.
Mhm.
Obviously, if we had an answer to that, then Li-Huei, Lauren, and I would all be out of jobs, and the field would be done, right? And it would be fantastic. But at least
Yeah.
one idea is that experience leaves some kind of physical, measurable change in the brain, and that recalling a memory is a way of revisiting that physical change that occurred during the experience itself, for example. So that is a result of neurons changing or brain cells changing over time. So brain cells will fire in particular patterns and change themselves over time. I like the idea of a symphony here where memory is like a symphony, and then all the instruments, they kind of tune to each other and play in different, either similar resonant frequencies or synchronize with each other, for example. When things get out of tune, something might go awry. When the whole thing goes out of tune, then we don't have a show anymore. So neurons do that when we learn. There's this kind of like fine tuning of the physical function and structure of memory of brain cells that are fine tuned over time as a memory is stabilized in the brain or as we recall it. But the key thing here is that this is a very dynamic process. Like there are these ideas that memories shuffle around in the brain, and that some brain cells are involved in certain memories for a time limited amount of time in this case too. So there is this kind of dynamism inherent to memory itself. So one last bit here is that when we recall a memory that's a day old versus 20 years old, it might not be the exact same set of brain cells that were active when we were first learning that experience that are reactivated 20 years later. So how is that baton handed off between different circuits and systems of the brain has befuddled neuroscientists for the past couple of decades. So on the one hand, that's a testament to how big of a problem it actually is. On the other hand, it gives us a job to do, right? So it can be win-win in that sense.
Li-Huei, did you want to say anything, or that was good enough?
That is good enough.
Okay, all right. Well, there's so many questions. We apologize for not being able to get to all of them. What a fantastic session. Thank you, Li-Huei, Steve, and Lauren, for this sharing of your knowledge and the discussion. Thank you also to Megan and Tammy and Kim, who helped organize and run the session. And thank you all for attending. Don't forget to check out the MIT Club of Boston website for information on this session as well as on future session. Bye-bye.
Thank you.
Good one, everybody.
Bye-bye.